Good morning. We are continuing our movement down or up the phyla of the animals. We are still in the multicellularity. Hopefully, we will always stick in the multicellularity, but we have to talk about new, different descriptive terms for this new phyla of Platyhelminthes, also known as flatworms. These flatworms involve something called an acoelomate, which means without a gap, a without. And we'll talk about what a coelom means in a little bit. There are three classes that we're going to talk about. Tubellaria, which is the only one that has free living organisms, and that means living outside of a host. Trematoda, which is by and large a parasite class, where we have liver flukes and other interesting creepy crawlies that live inside of us and other animals. And then finally, we have the Cystoda, sometimes seen with a spelling with a IA at the end. Which is the tapeworm class. Again, a flatworm, but in a very interesting way. So we have already talked about sponges, Porphyra phylum, being multicellular, but having no true tissues. Then we talked about the Cnidaria class, and we almost only talked about comb jellies, Cnidophoria, a little bit, but both of these phyla will only have two germ layers or two tissue layers, so they have true tissues and they're radially symmetrical. The Cnidaria also has stinging cells. So past this group, we get into a larger characterization of all these groups with whether or not they have a coelom. And this has to do with now having three tissue layers that all the tissues in the body start out from, called triploblastic. Blastic talking about the germ layers. So triplo versus diplo. So all the rest of the groups, all the rest of the phyla past this point, past jellyfish, will have three germ layers. Now, because of this, we're going to have to define them as what they have and what they do not have. Since they all have three germ layers, we have to talk about differential characteristics here. So we can talk about the ribbon worms. I'm not really going to talk about those too much. I'm going to talk about Platyhelminthes, the flatworms, where they do not have a coelom, also known as acoelomates. So we are still at the one or two-way digestion plan, where what comes in, the leftovers will come out through that same hole, versus what we will develop into later on, where we have phyla that has a tube within tube, a one-way digestive system. We are not at one-way yet. We have this two-way digestive system still. And sometimes we don't even have that in this group, in this phylum. So when we're talking about the differences between these animals that start out with three germ layers, we are talking about the ectoderm, the mesoderm, which is a middle dermal layer, and the endoderm. So ecto outside, endo inside, and then the mesoderm. So really, when we're thinking about these animals, even with their length, even though they might not have a full digestive system a one-way, they will have this tube within tube plan. Now, acoelomates just refers to a coelom. A coelom is a gap in between the endoderm and the mesoderm in this case, but at least a gap between the tissue layers. A true coelom is a gap within the mesoderm. This allows for expansion and growth of organs and development of organs, including blood vessels. So there is a large amount of animals that have a true coelom. where the mesoderm has a gap within itself, and the mesoderm enwraps and supports the endoderm, but allows it to grow. There is some shock resistance value for the liquid that is in here. So every time somebody gets hit in football or trips and falls down the stairs, they will not have a rupture of their spleen. Pseudocoelom is a fake coelom where you have a gap in between the endoderm and the mesoderm. It is not a true coelom, so pseudo, fake coelom. And we have roundworms in there, but the flatworms have no gap in between the mesoderm and the endoderm, but they still have 1, 2, 3 cell layers. So acoelomate is a defining characteristic of this group. You will need to know the differences between all of these up to and including drawing and labeling a figure. You should also know which groups have this characteristic. It is going to be important for separating them out later. So with the first class in here, this is the free living worms, Tubularia. It's fun to say. Let's talk more about what they don't have. They do not have a circulatory system. They have very few official systems at all, but their circulatory system is lacking. They have no blood system. They have no blood vessels, heart, whatever. They only work with getting their oxygen from the environment and releasing CO2 to the environment. They live in water. So they are thin enough so they can diffuse CO2 out and diffuse oxygen into all the tissues. They have a digestive system. It's two-way, and you can see it really well in this organelle or in this organism. They have an open pouch basically that has a little a searching suction, kind of like a vacuum cleaner hose that comes out. It's a pharynx that everts and feels around for food. And then it goes into this pouch with a whole bunch of little villi or openings along the entire length. That is the extent of its stomach and kind of like a cecum intestinal pouch, where they'll break down food individually in these areas and distribute it by, you guessed it, diffusion throughout the length. Any extra things that they cannot digest are put out through that same hole that is the mouth. So they have a very simple nerve net. They have some, it looks like googly eyes, cross-eyed googly eyes here, but these are light sensing pits. They also have these almost barbs on the end that will sense chemical differences as well as electrical differences, which is quite interesting. This is called cephalization. Even though there is not much there for ganglia, we have an organization of one end of the organism being the head with a little bit more nerve endings. This animal has a remarkable amount of regeneration. So if you cut it in two, it will regrow its other side. If you cut it down to here, you can get a two-headed flatworm. The one thing that these animals do have is an excretion system. And this is very interesting, being the first official system in the body. And I've talked about the excretion system while in that wonderful time we were actually together face to face. But here is that pharynx again. You can see all these protrusions or villi of its intestinal tract. Again, one or two-way digestion. So again, it goes in and out of the pharynx. This one has two splits of its intestine, if you will, down this way, and that's really how we define the different subclasses of the tubularia. Here's that pharynx that everts. It's kind of like a sock inverting and then feeling around for food. The auricle is there chemical sensing spots. A lot of their body is made up of reproductive organs, and they have this very crude neural net, lateral and longitudinal nerves, and that's about it. And then they have ganglion, just collections of neurons at the very um, front. What you can see in green underneath are the splits of the intestinal tract, and they have one that goes up to the top as well. So I mentioned that the flame, or that the one very advanced, or at least a full system for a function, is the excretion system. This animal lives in water the entire time, fresh water, so it has abundance of fresh water, and it does not want to dilute out all of the salt in its cells, so it has to get rid of the water permeating its body some way. It also needs to get rid of ammonia, which is the most basic nitrogen excretion release. So it has this set of tubes. It has this tube with a set of flagella here that waver back and forth to push all the liquid that's seeping in from the cells around it, and all the ammonia down this tube and out this very complex network and out into pores all over the place. So this is called a flame cell, and these excretion pores. The flame cell is called that way for the following reason. This is a flame cell, and what you're going to see is movement across the screen. One of the early microscopes that was developed looked at these flame cells, and all they could see is this wavering back and forth. It was nowhere near what we see here. And this wavering back and forth looked like a flickering candle flame, so they called it a flame cell. This cell will cause movement of water from the internal cells, or the cells around it, down the set of excretion tubes, and giving it a direction. And the direction will be out of the body, so it moves excess water and excretion products such as ammonia out. So it is actually well explained or named with this flame cell, and this is one of the characteristics I want you to know. The excretion system is one of the first that is fully developed, or developed to a way to, that is very important. So this idea of osmotic regulation, as well as excretion of these nitrogen byproducts, is one of the more important things an animal can have. So it has a two-way digestive system, basically a lot of reproduction and a lot of this excretion, this flame cells, and that's about it. Now, class Trematoda is definitely a parasitic organism, or set of organisms, and this is where we start talking about characteristics that help them live in a host. They have something called a holdfast device. And two of the types of animals that we talk about here are Clonarchus and Schistosoma. Clonarchus is the liver flukes, and I have some pictures of that. These are all parasitic, and mostly they cause agricultural and wild animals a lot of issues. But once in a while, they will affect humans. This is one of the reasons why I really like the Federal Food and Drug Administration. They make sure that we have a good amount of food and healthy amounts of food. Because of that, the life cycle of this animal is rather interesting. It starts out in the water, usually in snails. So a number of these embed into the snail tissue because it's soft and is in water as well as on land. And then it develops into Saccharia, depending on how you pronounce it. It'll embed itself in fish and things like that, and then the fish will be eaten by humans. If you cook it, it will be fine, but if you undercook it, so you do not kill the parasites, it will go badly for you. The animal, once it survives the trip through the stomach, will then get itself into the bloodstream and travel to the liver, sometimes going directly up the bile duct into the liver and embed itself there. It will live in a little cyst in the liver, taking up resources and damaging the liver over long periods of time. This is an example of a damaged liver. It is not the correct color. It is having spots on it. And when we cut into it, this is a good example of a very large moose liver. And this is a liver fluke with a capsule around it, kind of walling it in. This is not a healthy liver. This animal is probably dead because of the liver flukes, but look at the size of them. They're almost the size of a baseball. Some of these liver flukes can get quite large, while others are relatively small. Depends on the size of the animal. So class Trematota also has another animal called schistosoma. And this one will definitely infect humans. It is known to be in and around waters. We know it as swimmer's itch, but fear not, you can get swimmer's itch up in this climate and not have the full infection. Lucky you. This is a dioecious worm. Even though worms are spelled wrong here, this dioecious worm means that it has two sexes, the male and the female. The female has this groove that the male just lives in, but both of them have these suckers called holdfast devices that hold it in the intestine or bladder. They release thousands upon thousands of fertilized eggs, and they have this little tooth on them, which makes it very easy to identify what this organism is in fecal preps of this. I did this many times in the clinical micro rotation labs, is prep fecal samples and then identify what parasite is in there. A lot of the time this hook will actually damage the cell wall or the intestinal wall and cause it to bleed, sometimes causing a superinfection of the worms in the bloodstream. But if it gets defecated out into the waters, and some places actually use human feces as a fertilizer in these waters, it will hatch into basically a larva with cilia on it. It will actually embed into a snail's skin. It's really interesting because it touches this skin to embed, and it blows off its outer layer, kind of like armor it's getting rid of, and embeds into the skin. It lives there with asexual reproduction, and then emerges out as Cercaria larva. Basically, it has this flipper tail and a larval head in it. It will seek heat and go and embed in the first skin it has found, usually causing a rash. But then we get the infection that travels in the bloodstream and then eventually will seed itself in small intestine and bladder. This is also known in our neck of the woods or geographic location as swimmer's itch. Chemically untreated waterways that have ducks pooping in it, or waterfowl of some sort that migrate, will actually have this infection. Now, we do not get the full cycle of the infection because in this water, it's too cold for them to correctly develop, so these larvae are essentially dead. They can still heat seek and embed into our skin, causing a nasty rash, but other than that, we do not have the infection because the larva is dead. Lucky us. Here is an electron micrograph of the dioecious adult worms. There's the male sitting in that groove, and the female has the holdfast devices. This is the Cercaria larva, shown with Fitzy stain for this. The final class in this group is class Cystoidea, and this one has the I and the E, so there's a number of ways to spell this throughout the known world. This one does not have a mouth, even though it looks like it. It has absolutely no digestive tract, so this one is different from the other two, as in having absolutely no mouth. We know this as tapeworm or tenia. This animal gets its oxygen through diffusion. It releases its CO2 through diffusion. It gets its food by diffusion through its thick skin, if you will, and releases the ammonia through diffusion. It is surrounded by food in the intestinal tract. And yes, this is something that lives in your intestinal tract. Its head, if you will, it does not have a very well developed neural net at all, nothing like tubellaria. It doesn't actually have cephalization or a head with a neural link, or concentrated neural activity there. It has suckers in a set of four. You can see the other two on the other side that help hold it in the intestinal tract, and actually kind of grappling hooks with a sucker that will attach it to the intestinal wall. It will attach right in the upper part of the small intestine, the duodenum, and then allow its body to trail behind it. And you can see these segmentation starting that get into a much larger segment as they develop. But remember, this is a scolex, this is a holdfast device, and not a head. Here's another picture of it. This is the actual tapeworm. The scolex is here at this thin end, and you can see these segments start developing. Each of these segments is an egg factory, essentially. The only developed part of the tapeworm is a vast amount of reproductive abilities. Each of these segments contain thousands of fertilized eggs from itself or from other segments. So it's a hermaphrodite. Each of these segments, once they're developed, as we go down towards this end, it is not a tail, it will release or break off each of these segments, and it'll fall out in the fecal amount. You can have a super infection of this tapeworm that will get into the bloodstream, into the tissues, and even into the brain, which causes a life-threatening injury. Some people will take this as an extreme weight loss device or a way to suppress their allergy immune system. This is a very unsafe way to do it. So cook your food. I remember somebody had a fish tapeworm taken out of them. It was 9 foot long. It was on the Marshfield Clinic microparasite bench, and it sat there as their wall of weird. Somebody had been eating undercooked smoked fish, and I read that is somebody got drunk ice fishing and decided on a dare to eat a raw fish. That's, given my experience and understanding of ice fishing, a fairly common event. Apparently people get bored. Here's the life cycle of it. I do not like this scolex picture, but it is a scolex. It'll attach to the intestinal tract. It'll release eggs. Usually it is eaten by pigs. The pigs, because they root around in dirt and such, will get this super infection that will be even in the muscle. So you have to cook your meat, especially in pork. There's a number of parasites that will infect you from there. So it can infect your intestinal tract and live there. It can colonize multiple different areas, where it is shown by the lightning bolts. Here is how it usually lives. Here's the scolex embedded in the cell, the intestinal wall, and it trails behind in our large amount of intestines. These CAT scans are not a good sign. We have a cyst in the head here causing major trauma. And we have cysts throughout the brain in different areas, all because of uncooked pork. These are actually small worms that have been encapsulated but have caused some major issues with brain capacity. This person is, I, mm, I cannot see them other than dead by this infection. Each of these small white masses is a tapeworm embedded in their lung. This is not normal and usually ends with the death of the host, but this is a superinfection with tapeworms. Uh, this is the end of the first part of Platyhelminthes and nematodes. So this is all phylum Platyhelminthes.